Yeah, so I am a diatom ecologist, or sometimes I call myself a freshwater ecologist. I work on the taxonomy, ecology, and paleoecology, so the historical record of diatoms. And so I thought I'd just start out by giving you an introduction to these organisms and what they are all about. And sort of the short version would be that they are algae. They're unicellular algae. And I think all of you sound like you have a pretty big background in biology, and you probably are familiar with different kind of groups of organisms. Algae is not a phylogenetic classification. It's a just a scramble of things. It's like calling organisms bugs. We know that there's insects and crustaceans and all different categories. Algae is a similar term. It's just kind of a grab bag of that is applied. And more specifically, if we use a particular classification system, diatoms are eukaryotes, of course, versus prokaryotes. They're in the kingdom Chromista and the phylum Basilaриophyta. And within that phylum, there's three main classes: Basilaриophyceae, Coscinodiscophyceae, and Mediophyceae. And I'll come back to that a little bit later. The characteristics of the diatoms are their cell wall is made of silicon dioxide. Now, silicon is this incredibly abundant element in the Earth's crust, and diatoms have been able to take it in a dissolved form and incorporate it inside their cell and then excrete it to develop this cell wall. Diatoms are kind of a yellow golden in color, and it comes primarily from the types of chlorophyll. They're light harvesting pigments. Chlorophylls A, C, and fucoxanthin are the primary pigments of diatoms, and that's another important character. They make oils. They make these omega fatty acids, and they are a very important source of oils. They pass these oils up the food chain in the organisms that consume them. And so this is a good source of calories, and diatoms, so it makes them a particularly good source of energy versus something like the cyanobacteria, which are not particularly nutritious. And then really a diverse group. So the lower limit might be that there's 20,000 species. Some people propose that there are as many as 2 million species or more. And we are in that phase still of discovering the diversity of diatoms on the planet. So this diversity, this is a great example of the pigments. As you see this yellow, golden brown color, that is diatom pigment color. And it's something that is just so dramatic that even if you see these macroscopically, you can recognize them in a river where there's a growth on the bottom, or in the plankton of when there is a diatom bloom. So they're photosynthetic, and 20 to 40% of the photosynthesis on our planet is carried out by diatoms. So, and they are distributed in marine habitats, freshwaters, anywhere where there's light, nutrients, and occasional moisture. Diatoms also grow in soils, so they are ubiquitous, and in these different habitats are where these different species live. So here is one of the members of the Coscinodiscophyceae, the round diatoms. They basically come in sort of three sorts of characteristic shapes. And what we're looking at here, these are the silica cell walls. In this image, the cytoplasm has been oxidized away. Most of our taxonomy is based on the intricate structures of the silica cell wall. And so in a lot of the work that we do, we get rid of the inside of the cells and just look at the features of the outside. These are also, we characterize them by their symmetry, radial symmetry. There's a group, these are actually considered within the radial symmetrical group, but they have formed these elongate forms as another group. And finally, a group that has this characteristic slit that goes through the silica cell wall. This is a scanning electron micrograph of the surface of the exterior of the cell wall. And they have this, looks kind of like a break through the cell wall. This is called a raphe system, R-A-P-H-E. And diatoms that have this raphe system are capable of motility. So they live on surfaces and they can migrate through sands, through muds, and so these are the motile forms. And we also think that they're the most advanced. They have the most changes from their early ancestors. Now, one of the really interesting things about diatoms is that because of this silica cell wall, it constrains the cell size. The cells cannot expand. And so even back in the early days of microscopes here, 1896, Henry van Herk observed that over the lifespan of a diatom, the cells got progressively smaller until at some point the cell wall actually splits apart. And there's a lot of variations on the life cycle. We'll take a simple one. Haploid amorphous gametes emerge, fuse, form an oxospore, and elongate to get to their maximum size. These are the only organisms that we know that have this type of really kind of bizarre life cycle, that they get smaller over time, and it's the sexual part of the cycle that allows them to restore, is a size restoration process. And if we look at how that happens with diatoms, here's just a diagrammatic view of a parent cell. We call a cell a frustule. It's made up of two parts, a epivalve and a hypovalve. And when a cell divides under normal vegetative division, this parent cell passes on to one daughter, and that daughter makes a new half of the valve, indicated in red, which is smaller. And the other one with this hypovalve goes to the other daughter, and in it makes a smaller valve. So the vegetative cell division results in a characteristic decrease in size. And people worked this out quite a long time ago, figuring out the size of each of these halves of a diatom cell, these two it's like a petri plate that petri dish, two parts of the petri dish fit together, and that they get progressively smaller with each cell division. And so if we look at a whole diatom population, they actually start out big here in microns. And over time, the cells move, cell size, the valve length, moves to the left in terms of their distribution, and the size distribution of the population becomes more spread out. And this is a characteristic feature of diatom assemblages. Once they reach something like 40% of the maximum size range, different environmental factors like light, nutrients, temperature can stimulate the sexual cycle, and that those oxospores will be formed, and then there will be a very small number of cells at the maximum size range. And I don't know, I think this is just really an amazing and really fascinating feature of the diatoms, that they have this life cycle. And here's what it looks like. Here is a, we would call this the parental cell. In this case, as there's many different variations on this, these two parents have split. They're inside a mucilaginous envelope, and these are two oxospores, and they are elongating through a series of not flexible, but siliceous bands that allow the cell to reach its maximum size before it starts the vegetative cycle again. So these size and shape changes are vary across different species, and so then people who study diatoms need to understand that you know, we look at things in kind of a size series within a species. And that's part of understanding what a species is what's the change of morphology within that group as dictated by these patterns as being constrained by this external inorganic rigid cell wall. And these have also been shown to hold up in terms of each one of these groups here represents a different shape and size series within this group, the species Celophora pupula. And sequencing based on 18S, the ITS regions, and RBCL, which is a chloroplast gene, show that these hold together. Both the morphology and the gene sequences show that this is reflective of being, that valve shape is reflective of biological species. And so here's an example of a species that has a really very extreme size range, from these very young baby cells to the oldest of the generation of diatoms. And one sort of interesting thing is, if they are not, if they don't experience favorable conditions for cell regeneration, they can just divide into oblivion. And we have some records of actually documenting extinctions when cells fail to be able to regenerate their large size again. So we're pretty sure diatoms evolved in the oceans. And in the early, we don't have good fossil evidence of this. It's mostly based on molecular clock estimates. And part of the understanding is that in the early oceans, there was, a lot of, silica is this rich element on the Earth's crust weathering of rocks into the oceans, and dissolved silica concentrations becoming very high, and for some organisms even becoming toxic. Diatoms figured out how to take up dissolved silicious acid into a special organelle called the silica deposition vesicle and excrete it. And that turned out to be a good thing against organisms that are trying to, it had a rigid cell wall. So the evolution of the diatoms is tightly aligned with the Earth's history as well. So the concentrations of silica in the oceans, and also with the concentrations of CO2, and how those were aligned over time and where those populations developed. The radial centrics are primarily the marine species, the plesiomorphic or primitive form, the bipolar and multipolar centrics being more advanced, and then finally the raphid pennates, the ones that are long and thin, and the one I showed you that had a raphe system. So here's an example of a little bit of a moss growth and all this oxygen being generated by the diatoms. Look for that characteristic golden color, and this oxygen being trapped here in kind of the biofilm matrix. Diatoms produce more oxygen than even the rainforest. So they're really important in the global scheme of things. These are some diatoms that grow in sea ice. These are from Arctic sea ice. They grow in the brine channels in the floating pack ice. Here's some examples of here's a living cell. Again, we see the chloroplasts, light harvesting pigments here are in a actually diagnostic position, and lipids within the cell. They make lots of these lipids so that they can get through hard times, and they also get passed on to other organisms. There's the nucleus of the cell. This particular genus, Orthoceira, only grows in moist habitats, or what we call splash zones, along the edges of rivers and in waterfalls, sometimes in the bark of trees. So these species are, adapted to very specific conditions. Here in this scanning electron micrograph, we can see that these are all diatoms, all these things here, and they're growing epiphytically on the outside of another plant, probably a green algae. So these two have made a stalk, and so they're attached to a surface. These are line adenate, so oppressed to the side of this plant, and these have very little ability to move. They sort of are stuck there. These others here can actually have a raphe system and can move. And so what we're looking at here is almost like a forest. We see a three-dimensional structure of the understory, those species. This is a cocknease that grows early colonizer on the first part of the available substrate. Then some of these other forms come in later and build this really three-dimensional structure. So they're not all diatoms are not the same. They have very much different habitat preferences, growth preferences, nutrient preferences. And here is what This is an image of a stream. Here are some cobbles or rocks. And this is a diatom gomphonis that is growing in strands. So macroscopically, we can see these things. These colonies are large enough, and there they've got that characteristic golden brown color that you can recognize it, from the edge of the stream that this is a diatom. Here are some algal mats. These are from permanently frozen lakes in Antarctica. And these mats are a matrix of cyanobacteria and diatoms. And in this, these are really cool how these mats get here. I won't go into it too much, but they form on the bottom of the lake, and they actually melt their way up through the ice to the surface of the ice. These dry up and blow away, and they can disperse into other habitats. So these are just really amazing organisms. Here's this silica, is because it is inorganic. It's preserved in sediments in the bottom of lakes, in the bottom of oceans, and we can take samples from those sites in order to look at what the historical record was. And here we can see, this is from the genus Frustulia. It likes to be in kind of organic, rich areas. This is a Olicosyra, it's planktonic. This is an Agnanthidium that grows attached onto rocks. So here in this lake sediment, we have the history of what happened actually in the entire lake when these were deposited. And what that looks like in terms of data, this is a record from a lake that I worked on with my colleagues in Wyoming. And you have to adjust your XY axis here because paleocologists, we twist these around. Here's present day up here at 0, and here goes back 14,000 years. We can look at the colonization of this lake after this was from a glaciated watershed. And so how the diatom assemblages changed over time. And this is really a very powerful way to understand past environments by being able to see the diatoms that are there. And here we could look at the development of the watershed and how it changed over time. This is that same record where we characterized each of these groups into meroplankton. That's actually species that they can live on the bottom, but they get entrained in the plankton and they're just fine. How the planktonic species changed over time, whether they were growing on the bottom in the benthos, really started out, that was the main source of diatoms. Those that are able to live in soils, and those from rivers. So we can put that together as we piece together the understanding along with pine pollen and artemisia, which is a sage, and charcoal, looking at fires in this watershed. So diatoms are really very powerful that way to look back in the paleo record. This is a satellite image looking over the northern part of Scandinavia of a bloom of, this is both coccolithophores and diatoms together, showing just the pure scale of these blooms that can take place in nutrient rich habitat in places. And then sort of going back back in time further, um, you folks might have heard of diatomite, or probably more diatomaceous earth, as something that you put on your garden as an organic, to keep pests from eating your vegetables. But diatomaceous earth comes from diatoms, and they have been deposited over long periods of time. Most of the diatomites that are mined are from the Miocene, so anywhere from 10 to 18 million years ago, forming in the bottoms of lakes from large blooms, especially in the western US Great Basin area. And this also happens in, if you just think it's the ocean as well. And this is what this looks like. Here's a diatomite deposit for hundreds of meters, pure, solid diatoms from blooms that happened in the past. And there are many of these mines around the world that are mined for this algae. So that was kind of a quick ride through the diatoms. I wanted to end here with just a little pitch of the Diatoms of North America project, which is the project at diatomsorg, and how the group that I'm working with is what we're working to be able to develop this web resource so that people can learn more about these organisms. So I think I need to stop sharing right now. Actually, I wanted to get a chance if there's any questions that people have before I go any further on this website, or just to check in and see how everybody's doing.
So wait, actually, I had a question. First of all, thank you so much, Dr. Spaulding, because I've gone onto the diatoms.org all the time. Gotten a lot of information off of there. So thank you for that. Thank you for that resource. I guess I would like if you could speak to, I guess, like the applications of diatoms and how, research is moving forward with diatoms, using them as a resource for, I don't know, collecting oil and possibly creating biofuel and, all these great things that diatoms can be used for. So if you could talk a little bit about that.
Yeah, part of the reason that these algae have become well known and that we're able to know as much as we do is because of their applications to environmental problems, and as their use as indicators of condition. And that is, so I have a whole seminars on that too. But they are, really they came into being. Some of the Europeans found early on that diatom species are very picky about certain things. For example, pH ranges. We can characterize quite well the range of pH that most of these species live in. And that allows us, one, to reconstruct what the pH was in a lake. In lakes in the north, I mean, this is one of the great success stories, I think, of environmental science, is that diatoms provided a record, showed that lakes in the Northeast US and southern Canada had been acidified by sulfur released from coal-fired plants that were particularly rich in sulfur. The pH change, and this was through the 1960s and 70s, and the pH of these lakes changed so much, like from pH 7 to pH 3, and could be documented on a regional basis based on the diatoms and the sediment record. That data was useful in setting policy to change emissions. And that problem has largely gone away. There are scrubbers on power plants that prevent the acidic acid, maybe not some of the other things coming out, but that was something that has been a great success. We also use them to look at nutrient pollution. There are certain species that only grow in high nutrients and some that grow in low nutrients. So that's another way that we are able to use them both in modern senses and paleo. So yeah, that is a whole, the applications. And if anyone comes to our Diatom Web Academy, if you see those announcements on the webpage, they're largely directed at those applications that are done by the Environmental Protection Agency, U.S. Geological Survey, and states.
Thank you so much.
So I actually have a question related to the anthropogenic activities, and but specifically in the Antarctica. I know that you have studied diatoms there for quite a while, and I'd like to know if or how has it been studied the effects of human activity in the diatom communities in the Antarctica and the global warming and all this problem that we have right now. But for many people might be unaware that diatoms and many other microorganisms are changing because of our activities.
Yeah, that's right. And so we see that very much in terms of drought in places where we're reducing stream flow or reducing inflow into lakes. That changes the particularly the load of salts that are present. And so as a lake dries, it becomes more salty, and that shifts the assembly, the species that are there. And so I've worked in various places looking in that historical record in lakes. Of course, in rivers, we build essentially indices, so we can say for a given region, we expect this suite of species to be present. And if they're not, we note that as being an alteration, a human of human disturbance. And that involves modeling our expectations of species based on individual regions. And that's some work that I've been really proud to be involved in the past few years. I see someone asked a question about if a diatom has a rafe, essentially, does that mean that it has motility? And the answer is yes, that's right. And actually, the type of rafe that it has, they have varying amounts of motility from just a little bit, like they're kind of weak, they can only move a little, to being having a very well-developed rafe and being able to move very readily and move, some diatoms migrate with the tides up and down in sediments in tidal zones. So there's quite a range, to some that can just move a little tiny bit, to some that cover great distances.
So I have another question, a little bit different. And is it known that diatoms are part of the commensal or the microbiota of animals, like maybe sea animals, but other than that, is it actually well studied or not really?
That's an interesting question. I don't know of them being commensals. They do. There are some and again, this is specific habitats. There are some diatoms that only grow on the backs of turtles. There are some that only grow attached to whales. They position themselves and they get the stray nutrients that are, coming their way. They have cyanobacteria that live inside of them and will fix nitrogen for them. So, and the internal cyanobacteria divide along with the diatom cells, providing this means to grab nitrogen, even though they can't do it themselves. And we also know that diatoms have viruses. It was for a long time, people thought they might be the only organisms that did not, but it turns out people just had not looked that much. And so they've got their own virus load, just like everybody else.
I have a quick question. I'm not sure if it's a question or more of kind of a statement. So I've been reading some research on diatoms and specifically algae blooms, and there have been a lot of cases in areas of redistributed nutrients, especially in the case of hurricanes over bodies of water. You see like the red tides in Florida, and I see that a lot of the times these last for a very long period of time, and that the government systems in the area don't necessarily understand what is kind of going on. And I know you have a little bit of background working with government on this sort of topic. And like, what would you say is, in the societal sense, what, people are responsive to with this type of science?
Yeah, these harmful algal blooms are really a big problem right now, a major problem. And they occur across a number of different types of algae. You mentioned red tides, which are caused by dinoflagellates. We have blue-green cyanobacterial blooms. We also have, there are toxic diatoms as well, particularly in estuaries and marine zones. A genus called Pseudonytzia is responsible for the deaths of a number of marine mammals, particularly on the West Coast, California, Oregon, Washington, shutting down of lobster and crab fisheries because of these toxins. And there is definitely a strong relation to the amount of nutrients that are coming into estuaries as they come off of big river systems and changing oceans. And there's also a part that we don't understand. It's not entirely human. There's something else involved in a lot of these blooms. That's been something that's been, sort of surprising to me, in following some of the way things are as we're discovering these, because it turns out to be more complicated than we thought. And there are a lot of groups of algae that are throwing off these toxic blooms, and just the whole chemistry of how they do that and the nature of these toxins is really it's fascinating, and really how powerful they are to be able to influence other organisms.
Yes, I mean, extremely. They seem to upset an entire ecosystem, which is incredible for, such a small organism.
Right, and some of these, there are longer term dynamics that are part of the understanding. For example, in the Great Lakes, in Lake Erie, and particularly, there's been both blue-green algal blooms and diatom blooms that start. The diatom blooms, diatoms tend to do better in cold water temperatures and also in low light. And so some of these blooms initiate under ice before the ice is even melted. And they can set the stage for the rest of the nutrient dynamics that take place later in the season. So it's not just sort of, what are the conditions right now? It's what were the conditions over the past year that led to where we are now, and along those lines, the algal species that were there and how they altered the environment. For example, these blooms of diatoms can change the concentration of silica in the water such that they shut themselves down. They can consume so much silica that silica becomes limiting to growth. And so then that can pave the way for another algal group. Maybe it's green algae, or maybe it's the cyanobacteria to come in. So these dynamics are cross many different levels. They're related to inflows. They're related to predator-prey relationships. What are the zooplankton? And how are they consuming the algae? And really, how palatable are they? The green algae are something that is also a good food, but the cyanobacteria are just simply not very edible. So these are all very intertwined dynamics. Okay, Hardik, I had the same question, such as runoff from agricultural land into the water causing algal blooms. Yes. So when we think about that, we look at what is the size of the watershed. So in any particular part of a river or a lake, how much area is there upstream that contributed to this? What's happening in this body of water, and what's the nature of that watershed? How much? Some of the things we look at are how much of it is in agriculture, how much of it is in roads, how much is in urban areas. I've been working and looking at actually the diatoms as reflective of the amount of pavement within a watershed. We can track that very well, and we call it impervious surface, because that changes the nature of the chemistry and the stream channel. Even the stream channel is changed by that. So and yeah, they're very, very connected. Pesticides, we're finding diatoms are very affected by pesticides and herbicides, and we've also been looking at the pharmaceutical chemicals that are coming into the water. So far, diatoms don't seem like they are as sensitive to pharmaceuticals, but some of those other things that humans do to how we shape the land around us.
Very, very interesting. And I think there is a lot of things that are still there are a lot of things that we need to understand still, a lot of things to study. So I think we should, discuss a little bit about the database, the Diatom database. I know that many volunteers use it for their annotations, and so they're pretty familiar with it. So they might have some questions about the database.
Sure, I'll pull it up and share my screen, and I can show things, but feel free to fire questions away at me. I wanted to, if you haven't already figured this out, let you know about the how things are keyed in the search. So in the search box, you might have run across that there are a lot of different names for things, and you might have an old name. This is Agnanthes, minutissima, is actually an old name. And what we tried to do in here is to be able to help folks out with the being able to find something even if they're using an old name. And it might not be, so here's it, we came to the genera, we're searching species, the glossary and images. And usually the first return, the first element returned is going to show you that if you look at the basionym, or the original description, that this was first called Agnanthes minutissima in 1833, but it is now goes by a different name. That one's pretty simple, but there are some that the names have changed. And if you run across an old name, we've tried to put those in there and get you to the right place. And you've probably if you come across and come to the odd ecology section, we've given these characteristics of where this species is found. And this one's really cool too, because it lives in tubes. It makes its own mucilaginous tube. And here's all these guys living together in a tube, being surrounded by this blue-green bloom. And then here's some of these with the sexual reproduction of showing these parental cells with oxospores. They're just cool. Okay, so if you click on the highly motile button, you'll get a list of the other species that are characterized as being highly motile. And then I always find this is a little difficult to find. It's a little hidden because you don't automatically know this is there unless you go to it. But then you can access these other categories like this, nitrogen fixers. I mentioned that there's some that have cyanobacteria that live inside of them. And here's some of those that are nitrogen fixers.
I have a question regarding the synonyms. So when a taxonomist does the reclassification, is it based on the genetic information, like certain genes, or also new morphological characteristics? Is it okay to use the description, the morphological description, from the first person that described it, or when it changed the species name to a new genera, or new family, did it change also the morphological types, the morphological characteristics? Sorry.
No. So, and that's a great question about the nomenclature and how that works. Those names that we report here are based on what we call a type specimen. So a specimen is deposited in a museum and a name is tied to that specimen. And that never changes. So it's not going to change, even if the name changes, it's not going to change anything about the characteristics of that thing. Is that what you're getting at?
Yes. So because sometimes you will find articles, research articles, with the different synonyms of the same species. So some articles might include some information, some others might include another part of information. So is it okay to think about all those synonyms as the same as the same individual, as the same specimen?
Yeah, yes and no. I'm going to put it into the chat box here. There are things called homotypic synonyms, and there's also heterotypic synonyms. And the first ones, homotypic, are the exacts. They're referring to the exact same thing. Heterotypic are someone coming along and saying, I think that, okay, so here's the original, and they say, I think that this name is the same. So it's an opinion. And so if you look those up, that difference between homotypic and heterotypic, it's also, you might find it as nomenclatural versus subjective. And on this website, we only report the homotypic synonyms or the nomenclatural synonyms, just to be clear about that, and that they are the exact same thing. They have a different name for some reason in terms of trying to achieve a classification that represents evolutionary relationships. And our understanding is developing about that. So that's why it changes.
I'm sorry, I just had one quick question about motility. I see that the website characterized slightly motile or highly motile, and I'm assuming that's referring to the distance that they can travel. How far? Am I correct? And I guess like a second part to my question is also the different, I guess, I don't know, like, flagellum, is flagellum used as pilus, or do I guess diatoms, do they use these I don't know, their motility in that way to move further, so to speak? Like, yeah.
Those are really great questions. And and really, whether they're slightly motile, start with the first part, whether they're slightly motile or highly motile depends on how much of that rafe system there is. So some species, they just have it's a very abbreviated, and we don't know that if that's actually, well, we think pretty well that they evolved not having a rafe, and they started out making a little bit bigger ones, and then they got really big rafe systems. And so it's sort of the amount of distance that is a rafe is kind of defines as how well they can move with that. And then, flagella have largely been lost in diatoms. Their ancestors had flagella. Now some of the centric haploid gametes have flagella, but it's lost through the sort of the normal vegetative part of the life cycle. There are no flagella, so they're not moving by that. They're moving by actually, I mean, it's a fascinating system that they suspend some of these mucopolysaccharides through the RAFE system. They're attached to actin fibers within the cell, and then those actin fibers move back and forth and to allow the diatom to grow across the surface. I just posted a link. If you go to the news, under the news page, there's a new link for a site in Germany. A guy has done some fantastic videos of diatoms moving. And if you look at those, and they're really fun and really amazing at how they move. And he's really characterizing their speed and their direction of movement and how they can change forward and back, and sort of trying to understand that. Actually, there's some really great work. I don't think I really have it posted on here about the mechanics of that RAFE system.
So I have one last question regarding the secondary structures of diatoms. Are there any specific number of secondary structures, or are they so many and so diverse?
I guess I don't know what you mean by secondary structure.
I think she means like bristles, and I know some diatoms, they have bristles and they have spines. Yes, Rodwa, our diatom expert.
Yeah, so some species are for sort of far more elaborate than others, and they are all part of. I guess we don't use the term secondary structures. They're all part of there's part of the silica cell wall that's laid down, coupled with cell division. So it happens all at that point. And they're not they don't develop later. It has to be completed with the cell division. So yeah, there are spines that aid in cells being linked together. So joining of colonies. There are some species that we have something we call separation spines. That's spines that aid in breaking up chains and causing them to not be so long. There are some marine diatoms that have things that are they're like this, actually, like hands grasping each other, and allows chains to slide against each other. So there are a lot of elaborate structures, and their function can be in joining, sliding, separating. Many of them are just involved in buoyancy, just being able to stay floating, because silica is more dense than water. They need extra resistance to prevent settling out. Some of them have some of the other adaptations, whether they have resting stages, a cell that is able to perenniate or sit in the sediments and then germinate when conditions are good again. And those are really very group specific. Some make what we call internal valves, which is an adaptation against desiccation and drying, and are very characteristic. We can look at the history of a site by how many of these desiccation structures are present. Formation of, or the amount of mucilage that they produce, that is also another, sort of really key towards where they grow, of how much of a stalk they produce, and how developed the stalks are if they get very high in the canopy.
I'd like to thank you because I think this is really important for volunteers to understand this microbial group that many times it's kind of invisible to many of us, but it's of high importance in the environment and for us, simply the oxygen that we breathe. So I'd like to thank you again for being here, and thank you to all the volunteers who attended the talk, and thank you very much.
Well, yeah, thank you. I think your project is really terrific. I appreciate the work that you're doing, and please, I would love to hear back from you if you have specific questions or run into any issues with the website or things that are not there, or that you want to know further about diatoms. So I'm glad to make this connection with all of you. Thank you.